So yeah, so when you tried opening it, you would have done it like this. So you would have opened the file, and then in this case here, I think I have to see where the file is. Okay, I think the thumb drive is not connected. Let me see where the file is. It's supposed to be here. Yeah, so you will know where your files are, right? So it will be there.
GPSC.
Should I mute her from here? That's it. Yeah, so these were the files that so it will automatically show up, and so I can let's here if I do this, it will obviously it will I mean analyze it, and these are like relatively small. Yeah, let's go. Yes. Yeah, patches out here, right? And this is again the number of reads that have that particular Fresco. Yeah, right. And yeah, so that initial few errors are expected, just like any other PCR reaction. The initial part you might have some problems with this one, especially because we are talking about some adapters and all. Few of that remnants might be there, so perhaps that's why you might have that. Here you don't see a very smooth GC content because I've already given you a short, only a small fragment of the entire reads, right? So otherwise, generally you see this is the expected distribution of the GC content, which means that most of your reads will have a GC content that is close to 50% or around about that. But so you expect such a smooth distribution for your GC content. The next one is per. I can show you some results. Let's say this is after trimming. Not this one. Sample R2. Yeah. Sometimes you get some slight alteration. This was an exome, so you might have some biases also in that one right out here. So you can see there's slight different peak out here. But since it's an exome, I really won't worry too much about it. And also then the next thing I'm going to see is why am I getting that, right? So the next thing I'm going to see is this thing, N content. N content means the when you call something N means when you're not sure about what exactly is the nucleotide at a particular location, right? If it is not A, G, T, or C, you're not sure. If you have a lot of ambiguous sequences out there or very low quality read, then you call it an N. So here, generally, we're not seeing such ambiguous reads. Let me show you why we probably got that this error out here. Okay, one more thing. You can see out here. So here itself, you can see some ticks. So green tick means it has passed the quality filter. Cross means it has failed the quality, this one. And also you can see these symbols. These are warning symbols, which means that you can have a look at it. So here, when I'm looking at it, I can see there are a lot of there is a lot of G's out here. So homo polymer regions of G, which is present in a large count in this particular, particular read. So could be that is the reason why you are getting these things. Especially the GC content getting altered. Let me show you the next file. When I sort of removed that one, still it is there. So which means that it's okay. Let me show you whole genome, then it will be a little more clear. Where is whole genome? Trimmed. So it looks like this. Yeah, because even in the human genome, it's still smooth, and you can see something like this. So this would have been ideal, and you know that it is peaking at around 40%. So that is what it's speaking, which is expected for the human genome. So in the human genome, you see a slight lower number of GC bases compared to A and T. So that is about that. Suppose if you're finding a very large peak, very much off from this distribution, then you think of some other foreign DNA or some sort of a contamination there. Okay. Generally, yeah, this is about FastQC. So like I mentioned, so we can have we can have a look at the reads before and after trimming. Let me at this point of time show you the command that is used. So I remember when I told you the tool that is used for trimming. Let me show you. I made an infographic. Actually, this is generated by NotebookLM. It gives the overall workflow, whatever I'm going to tell you, but there are some spelling mistakes. I don't know why when it created, so instead of adapter and all, but it is good. It's a good enough visualization, I presume. So this is what we right now did. We used FastQC to see the raw read quality, right, using FastQC. And then I told you that we are going to clean up those reads. So how are we going to clean up those reads? Okay, so in this case, I am going to, I told you I'm going to use a tool, and the name of the tool is known as Trim-O-Matic. This is how the command will look like. I'll open it in a text file. So this is a command, and let me zoom it for you. You don't have to worry about this command as such, but this is how when you type in a command in a terminal, each software will have its own particular format and all. But this is how the command will look like. Or I can even, I'll show you one more picture. Just a minute. Commands. Yeah. This looks a little scary, but it's okay. I mean, it's actually simple. I mean, I've typed it for my information only, so that's why it looks a little too crowded. But this is how we actually run a Trim-O-Matic command. Okay, so Trim-O-Matic is something that is written in Java. So when you run the command, you type Java, and then these are terminologies that are associated with Java, this part. So Java and jar are all related to Java. Basically, what you're telling is, I'm going to use this particular software. So you can imagine, just like you have a software that is installed in your computer to open a file like Microsoft Word. So if you want to open a Word file, what do? You either you can do open with Microsoft Word. Similarly, something like this. So here I am telling that terminal that I'm going to open or execute this particular command called Trim-O-Matic out here. Okay, so this is where it is located in my computer. That is why this is pretty long. So this is the folder in which Trimmomatic is located. And this one, PE stands for paired end reads, okay? Because Trimmomatic can work with single end reads also, paired end reads also. And so when you say PE, it is going to work on paired end reads. What do you mean by threads? Threads are something that is related to your computer. I mean, if I put it in a very crude way, you might have heard of something like your processor, right? You have i3, i5, i7, let's say in Intel i9, also in Intel. Similarly, in your Snapdragon, you will have different processors. In Apple, you have M4, M5, like that. So when you say threads, thread means, how many times you can parallelly run this process is basically what is called a thread. So when I'm saying 12 threads, it means that, it's going to do this process, split it into 12 and do this process parallelly, so that, something otherwise that, for example, if it is taking 120 minutes, when you are actually doing it parallelly, it will proportionately take very less time. So that is why many times we say that when you work with these files, if you have a good computer with a good processor, which means, it will have more threads. Generally, when they use the term i7, I think it has 8 threads or 8 cores basically, and like that. So those are the numbers, and then you can have more when you work on servers and all. So here we have I'm using 12 threads also, so that it gets parallel. So this term will come frequently in multiple steps when we use this term threads, for example. And then now this encoding also. FRET33 refers to that particular quality score encoding. So when we're using FRET scores, 33 means you're using a particular. Remember those alphabets F and G and all those things. So that particular encoding is known as FRET33. So we use that also. And then what we do, then we tell that then your input material is, let me show you here the same thing. So now this part is clear. So you're using Trimmomatic, this command, thread12, FRET33. Now I'm going to give my first read 1 as the first input. Then this is the read 2 of that corresponding sample 1. So I can see there is sample 1 here. This is sample 1 again, but here it is read 1, here it is read 2. So those are my input files. What am I going to get? I'm going to get my output files. So I have to name them. I have to tell the software what it should name my output file. So this is paired read 1. So it will do all the quality scores, and here also I have another file which is called unpaired read 1. Then I have a paired read 2. I have an unpaired read 2. What do you mean by this paired and unpaired? You're going to do that read 1 and read 2, right? You're going to do the read 1 as well as read 2. So, what if your read 1 meets the quality criteria, but somehow your read 2 is dropped? Okay, that's possible because your read 2 came from a slightly bad region of your sequencer, let's say, and so read 1 is retained. Read 2 is it is having low quality. It means that you cannot. It gets filtered by this tool. So what is the problem there? Then now you have one unpaired read 1, right? Because now the read 2 is dropped in that process. So all those unpaired reads will go into this unpaired file. So unpaired read 1, and you also have an unpaired read 2 as well. So this is basically the file format. So you have your input files, you have your output files. Output files are again divided into paired and unpaired ones. And then we use some terminologies. So we tell them, for example, let me show you this part, average quality. So what should be the mean FRET score of the particular read for it to be retained? Here I am using a cutoff of 25. So if the read is having a cutoff score of 25 or more, it will be retained. Anything that is less than 25, it will be dropped. So that is the idea. And I am also using another terminology out here, which is known as minimal length, min length. So it means that the minimum length of the reads that I'm going to retain here is 70. Anything less than 70 will be deleted. Now see, I am also using a term which is called sliding window. What do you mean by sliding window? I can just show you what I mean by sliding window, something with the help of an example. So imagine my sequence is like this. Okay, I have a sequence like this. What do you mean by sliding window? Here it is written 5 and 20. So what do you mean by that? It means that, I'm going to look at the first 5 nucleotides. The quality score will be calculated. If it is more than 20, it will be retained. Then I go to the next 5, right? I slide along 1, 2, 3, 4, 5. The next 5, again, I look at the quality score. If it is more than 20, it is retained. At any point of time, if it is less than 20, then that read will be dropped. Okay, so that is how we do this particular process. So this is also to make sure that even locally also you get a good quality score, even in local regions, and overall you get a good quality score. 20 is again the Phred score. So I go for 5 nucleotides. If the Phred score is more than 20, it is retained. Yeah, right. 5 and 20. You can alter it. These are some standard formats. So instead of 5 and 20, I can put 6 and 30. Absolutely no problem. But these are some default formats out here. So that is something. Also, if you want to remove the adapters, you have to. You can tell the software what are the adapters that are used during the sequencing, right? So what are the adapters that are being used during sequencing? So this file, this is given by Illumina itself. So this is one FASTA file. Now all of you know what a FASTA file is, right? We have already seen what a FASTA file is yesterday. So let me show you what exactly this file is, just for some clarity. Okay, what is happening is, actually my mouse is okay now. Yeah, so this is a FASTA file. I can open a FASTA file with text editor. Can you see out here? So these are the sequences that are present in the adapters for paired and read. So this is my forward primer for the paired and read one, and this is my other primer for the paired and read. So yeah. Okay, so this is there, and so what will happen whenever Trigonometric finds out that this sequence is there, it will clip it off. It will clip and remove it off. Okay, so this is basically how it does adapter. Now, for example, in your particular protocol, if you have some other sequences that you want to remove, you can put it here as a next line. So you know that your FASTA will have a header and a sequence, right? So you can put something. For example, my custom, if I want to remove something else also, then what I do, I can put custom. For example, in the previous scenario, you saw that poly G region, right? So you can put something like this. So if you put it like this, it will remove those poly Gs also like that. Okay. So it's all up to you what you want to remove and what you want to retain and all those. But this is how you do a trimming altogether. And also, yeah. So this is how basically you do this. Here actually, you can see after I have executed this command, this is how it goes. So I have given 7500 read pairs, the file that you had, right? So you can check your FastQC data also out here. So your input sample, where is that one? This one. 7500 sequences were there, right? In your input, the one before trimming. That same number has been identified by this one as well. So you started with 7500. Of that 7500, 111 were dropped as such, completely removed, because their qualities were really bad. and which also resulted in some of them, they had only a forward one surviving, so only 161 had only their forward read surviving, 87 had only their reverse read surviving, like that. So what is of our utility is this number. Wait a minute. Somehow, just a minute. Yeah, so this number is more important. You know that after trimming, if you consider the pairs, most of your reads are retained. A few of them were dropped because of multiple issues, but most of your reads are retained. So this is basically what you do in trimming. And again, after trimming, we do one more round of QC. Now you know why I've written it as paired file, right? Because all your unpaired ones are now removed. You have only, so whatever files are there in my read one, there is a corresponding file in read two, which has also passed the quality filter and which is also adapter free ideally. So this is basically your read one and read two in pairs. The next thing that you're going to do is a very critical step out here. And that critical step is when you're going to align your read to the reference genome. You're going to align your reads to the reference genome. This is done using a particular step which is known as alignment. Alignment. So alignment means your reads are gonna get aligned to your reference genome. Now you know that because we have, let me open the PowerPoint as well. Get the PowerPoint. Good, yeah. Because we know that our reads are now all in pairs. So we know that these reads will have to be aligned accordingly, right? So it will be aligned in the form of pairs only, right? Yeah. So now you can actually align them with the help of your forward read as well as your reverse read onto the reference human genome. So your reference human genome is known as human genome version 38. That is the current version. There was one more version which came after that version 38. So the current version is, it's also known as T2T. T2T. Okay, it means a telomere to telomere. So that is the full version, which is actually much more comprehensive than HG38. But we don't use it because most of our tools are still not updated to use the ultra latest version of your genome. So your human genome 38, the current version that we commonly use, it came in 2013. Okay, but the other version that we are going to use, that is a recent addition. I think it came in 2023, and so that is known as T2T. Okay, let me show you. Simple. If you go to any genome browser where you want to visualize your genomes or look at your genes or something, so you get to see some number. So for example, this one, this is here. It is clearly written. Sorry, let me open it and show it to you. Yeah, can you see it here? So human GRCH 38. So this 38, that is basically the current version. The previous version is also known as HG19, which is also known as GRCH 37. This is an older version. Now what I'm trying to say is there is one more version out here, which has come after an update of this one, that is known as telomere to telomere, T to T version, which we are not going to use for any of our analysis as of now, but in future we might be using them. Okay. All right, so what are we going to do here in this process is alignment. So what happens in alignment? So you have these reads and you're going to align it to your reference human genome. How does your reference in human genome look like? I can just show you how exactly this will look like. For example, this is the file, and so can you see you start with, so it's a faster file again, so you can see it is starting with chromosome 1, obviously. And in the chromosome 1, you start with the telomeric region, right? You start with one end of the chromosome 1, a telomeric region, but a telomeric region, you're not really sure about the sequence because it is a repetitive sequence and you don't know about the number of repeats or anything like that for sure with the previous sequencing technology. So you write it as N, and then you have some regions out here, some sequences out here. And so you can see, so this is how the reference human file will look like. So you know that in which location, suppose you can count this, right? You can count it from one end, and you can know what exactly is the position of each of these nucleotides right out here. So chromosome 1, this is a position in the genome. So you can get that information. Similarly, you can keep on scrolling like this. So after some point of time, you will reach chromosome 2, chromosome 3, like that. You also have your mitochondrial chromosome and chromosome X and Y and everything in this particular file. So the tool that we use for alignment, there are again multiple tools to do this. But the tool that we are going to use in this particular process is called BWA-MEM. BWA-MEM. So what do you mean by that? Okay, what do you mean by BWA-MEM? It refers to the name of an aligner. So this is the terminology, BWA. That's the name of the aligner, okay? Burrow-Wheeler's aligner. Okay, let me explain something to you. What exactly happens when you use a Burrow-Wheeler aligner? Just a little bit of theory. I won't take a long time. Can you imagine this process? Can you imagine this alignment process? What exactly you're trying to do during your alignment? You have a, let's say, 150 base pair read. And what are you going to do now? You have this 150 base pair read. Imagine you have millions of such reads, right? And you're going to put this 150, you have to align it to your reference genome, which is 3 billion bases long. So you have a 150 base pair fragment. Now you want to put it to somewhere in your genome, and the genome is 3 billion bases long. How computationally intensive this is going to be, right? Putting that 150 across, along that entire genome. Now you imagine that same thing happening for millions of fragments, because you don't have only 150, single 150 file, you have millions of 150 base pair fragments. So now you have millions of 150 base pair fragments. Each of them should be mapped to a particular region in the genome. This is computationally very intensive. To make that process easy, there are different algorithms to help that. One thing that is usually done is that, you sort your human genome sequence, you break down your human genome sequence. The 3 billion base pairs are, let's say, artificially broken down into small fragments, and you can keep all the similar fragments together. Does it make sense? So I'll repeat it. So you have that 3 billion base pair long human genome. You can chop it up into small manageable size fragments. I'm telling about the reference genome. Okay, you can computationally split it into smaller fragments, and you can keep all the similar or relatively similar looking fragments into different clusters or groups. That is one thing that is done. The next thing that is usually done, now you take a read. Now this read is 150 base pairs in length. Now, if you are to put that entire 150 and compare it with this region, that is not going to work, because I mean it will work, but it will take a lot of time. So what you do, you take some small seed regions. You create some seed regions in the reads, and now you see if the seed region is where exactly it is going to match to your chopped up and ordered reference sequence. So let's say here you can see an AGC is coming and matching. So there is a seed region which has a match. Now what you do, now you look at either side of the seed region if the other things are also matching. If it is matching, then you keep on extending and align it. If it is not matching, you try to match it somewhere else. So this is exactly what happens during sequencing when you are using this Burrow-Wheeler's aligner. So two concepts out here. Your reference genome is usually split into multiple manageable sized fragments which are arranged based on their sequence similarity. And your reads will have a seed region, which will go and find the corresponding region in the reference, and then it will extend and see whether the map, whether it is getting mapped or not. That is why it is computationally really intensive, and this is one step where you require a good computer. Because it takes a lot of computational efficiency for this to be done in a good amount of time. Okay. I remember when I was doing this using a different aligner, it used to take an exam used to take something like 4 hours or round about that. Because my processor was very minimal, but now these days it happens in a few minutes with large files because the system has been upgraded. Any questions there regarding alignment? Yes, you can do that. Hmm. Right. Yeah. Yeah. Right. So you don't have to do any of this. So let me show you how do we do this again. Right. It got stuck. Actually, my iPad is also on, and my mouse is somehow skipping one to my iPad. So yeah, right. So we don't do all those. I mean, I'm just telling all that the software will do all those for you. All you have to do is give the command, and the software will do that particular process. Okay. So generally when yeah, this is called a pipeline. So yeah, so now you see my pipeline. I can show you this actually. So this is my pipeline. Where is it? This is my pipeline. So what is a pipeline? Pipeline is nothing. So you already saw a couple of steps in the pipeline, right? First QC, trimming, another first QC, alignment. So you already done 4 steps in the pipeline. Right. Okay. So this one, how do we do this one? Yeah, this is how we run MEM. Again, I have done something a little tricky part, but if we want to run an MEM, extract read group. Let me show you the command here only. That's easy. WMEM. Basically, this is it. It's pretty simple. Yeah, this one. Can you see that one? That's a command. Oh, wait, when I zoomed it, it disappeared somewhere. Where did it go? Illumina paired and reads longer than this one. Yeah, this one. Visible? Okay. So it's pretty simple. You're telling, so just like in the previous step, you used Trimmomatic. So here we are using BWA, and we use something called an MEM. Actually, I forgot what is a. I think it's something minimal essential. I mean, okay, I won't guess it. I'll tell you tomorrow what exactly that means. But there are multiple algorithms even in BWS. This is the algorithm that is preferred. So you can see MEM is one. This one is a different algorithm. So in BW itself, you can have different algorithms. So we are using this particular algorithm. What is this file? This is your reference FASTA file, the one which I showed you, right? That HZ38 one. That is your reference FASTA file. What is this one? This is your read one, FASTQ. So you can write FASTQ as FASTQ. That is one way of writing it. You can also write it as F.Q. Both will work. Okay. So here, when I gave you the file, it was written as FASTQ. I can also write it as .fq. Both will work. So here you can see read one. This is read two, and this arrow means it is going to write it to an output file, and that output file is known as a SAM file. SAM file. So you already know about FASTQ file. You already know about FASTA file. Now I'm going to introduce you to a third type of file that is used in informatics, which is known as a SAM file. What do you mean by this? Okay, maximum extract match. Okay, fine. Right. So MEM. So for all our workflows, we use this BWMEM only, and because you can see, because this is a one for Illumina paired and long read, more than 70 base pairs. We use that. If it is any other value, then we may have to go for something else also. Right. So read one, read two. So you have both your paired and reads. You also have the reference, and you're going to write the output to an output file, which is known as a SAM file. What do you mean by a SAM file? SAM stands for Sequence Alignment Map. It's an aligned file, which means that, I'll show you a SAM file. Let me show you a SAM file. I created one SAM file for you. So you can see the SAM file. Again, SAM files are very large, okay? So can you see the size is 28 GB out here. That is why, because, it has a lot of information, right? So now it has information about your reference genome. It has also information about where all your reads are going to map against your reference genome. So let me open and show a SAM file for you. So this, yeah. So this is how a SAM file will look like. Every SAM file will start with a header region. So these are my headers. So what will be there in the header region? It will have all those chromosomal locations which were used for mapping or which were present in the reference file, or all those chromosomal locations that were present in the reference file. So if you look at it, you can see chromosome 1, then it goes actually alphabetically. So, 1 followed by 10, 11, like that. And after 1 is all done, then you get 2, 3, and all like that. And so here you can see there are two things out here which you can notice. For example, when it comes to chromosome 9, for example, this is chromosome 9, right? So this is chromosome 9. It has a length of 138. This is a particular length of this many nucleotides are there in chromosome 9, 138, 394, 717. So there's that many nucleotides are there in bases are there in chromosome 9. Similarly, you can see the length of chromosome 6, 7, 8, like that. Now you see, apart from that chromosome 9, so this is the main chromosome 9, you also see some additional sequences out here. For example, you can see chromosome 9 with some extra terminology, a version, etc. out here. So what are these? Okay, because this is what I was telling you. When we actually created our human genome, it was limited. It was made up of the genome of only, I think, 5 or 6 individuals altogether, which was merged and combined. But over the years, with a lot of sequencing studies, population sequencing data, we know that, let's say, a European genome of a European will have a lot more variation. I mean, it will have a lot of dissimilarity compared to a genome of an African or genome of an American or a black, African American or an Indian, like that. So what happened was, like, to include all those variations in the human genome, they started adding information about that variations also in the reference human genome. So this is what makes different versions of your human genome. So I told you already have something like HG19, human genome 19. When they upgraded to HG38, a lot of these things got added. So what are these? These are nothing but we use the term CONTIGs, CONTIG, CONTIGs. You might have heard of that when you read about human genome sequences. So these are nothing but some additional sequences which will explain the population diversity in the genome. So you see that for most of your chromosomes like that. So you will have, let's say, chromosome 14, and then you have a region of chromosome, a lot of regions of chromosome 14, which can account for some variations. And apart from this, you also have some sequences that you cannot confidently map to any particular chromosome. For example, you take centromere sequences, you take telomere sequences, you cannot for sure tell that this is coming from a particular chromosome. So such many sequences, you put it under chromosome unknown. So you don't know where exactly it is coming from, right? So that is also there. What else should I show you here? Yeah, I can see the mitochondrial the mitochondrial DNA out here. We always learn it. It is 16,569 bases, 16 kb. So you can see the mitochondrial DNA sequence also out here. And similarly, finally, you can see your X chromosome and Y chromosome sequences also out here. So that is the header. This is common for all the files. Yes, sir. Hmm. Oh, no, no. Actually, this is something that is coming from your, this is coming from your reference genome, but there also you cannot tell for sure where exactly it is coming from. Either way, I mean, in your say, ultimately your genome is also an assembly from different sequence runs, right? So we cannot for sure tell that where exactly these are coming from, because the reads are only 150, and here this 150 is also not good enough for that, or even your 300 is not good enough for that. That is why we have an update to the human genome. Okay, because now in the current version, which is based on long read sequencing, so you don't have many such ambiguous regions. Most of your genome, they say that 99.3 percentage of your human genome is now mappable, and you have the exact sequence for 99.3 percentage of your genome. When we say human genome project was complete, I think it was somewhat like 92 percentage of the genome that was complete. Even though we told that we got the entire human sequence, that was not correct, because especially when it comes to repetitive regions and such ambiguous regions, these were there. Because of that, 8 percentage of them were still ambiguous. But with long read, now that 8 percentage has been reduced to 0.8 or 0.7 percentage. We got a lot of further information nowadays. So ideally speaking, which reference should we use? We should use telomere to telomere. But we cannot use it because all our databases that we are going to use further on from here later for all the filtering and everything are based on 38 still. So that is a problem. So if eventually if people are going to build databases based on the latest genome build, then we can use the latest genome build also. Okay. All right. So the header also will tell you. I mean, you can ignore this part. The header also will tell you what commands and all we used for this particular process. And let's come to the actual part. So that apart from the header, these are the actual sequences that are out here. Okay, no, not exactly that line. Let me take you. SAM file. Yeah, SAM file. Yes. Let me take a particular read out here. No, I mean, I can open it in Notepad. I mean, these are all like in the text encoding only, but this is, since it's a 28 GB file, my Notepad will crash if I try to open it in my Notepad. So that is where you work with terminal. So in terminals, you can just open it just like that. And if you want to say many a times, what happens is sometimes you get some errors because your header has some issues or something. So you may have to delete a few lines, 10 lines or something. So if I try to do that in Word or something, then it's not possible. But in command line, I can do it with a simple command. Just use one line and do that. That's why we use command lines for this purpose. Okay. Now you see this file. Actually, when you get an output SAM file, I mean, so here you can see this one, right? Can you see an ordered arrangement out here? For example, chromosome 1, 14507, chromosome 1, 14516, again another read, 14516, 14575. So it's in that particular order, right? It's going right from chromosome 1 onwards, and it's in the increasing order of the genomic position. This is because I have already done something called the sorting. So you sort the file according to the genomic loci, chromosome wise, and the position wise. So that is also another step. I haven't mentioned it here in the pipeline, but generally when we do that, so you get a SAM file at this stage, and you also do a sorting of the SAM file. Okay. I can also show you okay. Yeah. So what am I trying to tell you here? Right. So this is basically structure. So can you see? So I have a sequence here. So this is my sequence, right? This is my sequence. Are you able to see the one that I'm highlighting? Oh, wait, I lost it. Yeah, I tried. That is okay. So here, this one, that is your sequence. And yeah, so that is your sequence. What is this? That is a Phred score. Yes. Okay. And then you get some other info. So you know that this is a file, this is a quality. It is aligning to a particular location, chromosome 1 at this particular location. So now you know this is exactly what happens when you do that alignment. Okay, and then what else is there? Mostly, there's one more important thing that we can have look at. So this read will go from that entire location, right? So chromosome 1. Yeah, exactly. Then it will go. I mean, it varies actually. So you can see a read length out here, right? So 117. Yeah, yeah. So that is 117. You also get one more information out here. It says M. Okay, M 117. It's written M. What do you mean by that M? That is a very important concept. So let me again. I don't have a particular slide for that. So let me just show you what do you mean by that. I don't know. My chat GPT always calls me madam. Okay. So it's biased somehow. Uh, yeah, that is called a cigar score. A cigar score means that M means you have a perfect match. Okay, so I'll show you some examples. Uh, so I am typing it as, let's say, 150 M, right? So 150 M means, yeah. So 150 bases are there, and these are aligned actually to that particular location. This is called a cigar string. I'll show you something else also later when we are visualizing, but just remember you have something called a cigar string right out there. And you also get some thing about quality. Let me show you what is the next step. So this is basically what is called a SAM file. So where we have that particular alignment that is happening. So this is called SAM file. What is the problem with SAM file? The problem is this. The problem is the size. Okay. Problem is size. It's a huge file. So I mean, if you are sequencing a large number of genomes, or I mean, this is for exome. Okay, this 28 GB. If I'm, yeah, this is exome. If I want to show the SAM file for, I mean, I don't even have a SAM file for my genome. I'll give you a comparison. Okay, so what do? You want to compress the SAM files because it's very difficult to store them. That is only disadvantage because it's human readable and you cannot. I mean, it takes a lot of space. So instead of storing anything as a SAM file, what we generally do, we store a binary version of that. We convert it into a binary version, and this binary version will not be human readable. Okay, we cannot read it. I can show you a BAM file. Let's say this is a BAM file. It's the terminal is already giving a warning. It's a binary file. You will not be able to see it, but if you want to see it, oh wait, what happened? Yeah, this is how it looks like. Does it make any sense? It's all jargon. I don't. I really don't. I mean, I do remember MS-DOS, starting with MS-DOS, but I don't MKDIR and all this, but I don't remember that part.
Yes. When you find this one, sir, right? Absolutely.
But this is a file, okay? So you cannot read it, but you can read the SAM file. So what is the advantage of this one? What is the advantage of this one? Now you see the size of that. That will be something like this, 2.89 GB. So it's like a more than 10 times compression when you do that.
Okay? Zipping?
Sir?
Zipping of the file?
Almost something like zipping. So zipping also is used for compression. This is a slightly different manner you convert it into a binary file. Zipping itself will not give you this much of compression usually. I'll give you an example. I mean, another instance why exactly we are doing this. For example, see, this is a BAM file. It is already 2.89, so almost 10 times. So now you see my BAM file. When I'm doing a whole genome, my BAM file was something like this. 70 GB is a BAM file. So BAM stands for SAM file. Yeah, right. So, when I'm trying to create this, I don't even generate a BAM SAM file in the sense, I mean, when you're working with these codes, you can take the previous input and immediately convert it into a BAM file so that it doesn't even get stored in my system. Whenever, as long as it's getting generated, it's getting converted to a BAM file itself. So that is how we try to save space because, I mean, especially when you're working with servers and these days with cloud computing, you have to pay for space also, right? So we always work with compressed file formats because of that particular reason. BAM stands for binary alignment map. So sequence alignment map is SAM, binary alignment is BAM. Now when I say BAM file, it's pretty, it's pretty clear what exactly that means. It's an alignment file, which will have all those information which I showed you, but it's in a binary format. So it has the alignment information as well. And in this case, I have also sorted it, coordinated. You have to manually do it. You are also sorting it based on your genomic coordinates. Now we do the one more important step. Okay, this is known as marking of the duplicates. This is known as marking of the duplicates. Let me tell you why this is important straight away. Let me see if I can show you one example so that I can show you this one. Okay, let's get it. So just to revise, by the time the tool opens, so we are here. So we have done the alignment, and we have done the we have generated a SAM file. Now the next thing that we generally do, what are you going to do with that SAM file? We did two things with that SAM file. One, we sorted it as per our coordinates, right? We sorted it as per our coordinates, and the second thing, we converted it into a BAM file. So this is done. Again, you can use so many tools to do that, but this is done using a tool that is known as SAM tools. I was mentioning it here because it's not clearly written here, but we use a tool called SAM tools to do these two things. Okay, so you can see this is a command that we generally use for SAM tools. So here again, now this terminology should be a little bit more familiar. So if I want to do this in the command line, what do I type here? I just type something like this, SAM tools. So that is the name of the tool which I'm going to use. What am I going to do? I'm going to do a sorting Okay. This number here for this particular tool, this number again means the number of threads. Okay. In the previous one, we wrote it as T. But here, this is again the number of threads. It means that, it's going to use 28 threads. It's going to do that parallel processing using 28 different sections of your computer. And you give your input file name. So this is my input, this is my input file name, paired file, SAM. This is my output file name, the sorted BAM. So it will do both the sorting as well as the conversion into a BAM using this one. So this is called SAM tools. So SAM tools, as the name suggests, it is again a free tool that we get, which is downloadable. And this is used for a lot of purposes, especially to manipulate these this SAM files. Okay, so I think. So that is SAM tools. Okay, this tool it is used for a lot of purposes out here. Okay. Again, these tools are again for Linux and Unix based systems. You cannot install it in Windows as far as I remember. I know. So you won't be able to use your Windows system purely for this. But if you have your Linux installed in Windows, you can still install SAM tools and all other tools that we are discussing here. Okay, so the next step that you are going to do is something that is known as marking the duplicates. Something happened. Wait. Sorry. I'm trying to open a tool, which let me find out where exactly that one is. Yeah, this one. I think my Java is to be updated. That's why it's probably not opening. It's not opening. Okay, let me show that to you in a slightly different context. Since I don't have that file there, I'll show you a picture. Then it will still serve the purpose. I'll show you a picture from the cases only. Let's take this one and see if we have IGV written. Okay. Out here. I will give you instructions how to install this software. So this is how we can actually visualize our aligned reads. You get a direct visualization of your aligned reads. You have a tool which can be downloaded and installed in all the systems, including Windows systems. This is known as IGV or Integrative Genomics Viewer. Integrative Genomics Viewer. What do you see here? Okay, if you have a look at it here, I mean, I'll explain this in detail later, but I'm just going to tell you what is the importance of that duplicate removal. So can you see each of these blocks right out here, right? Each of these blocks, it is a read. So it can be a forward read or a reverse read. So you can see the directionality of these arrows. So here you can see this one is in one direction, this one is in another direction. This is ideally a forward read. This is a reverse read, another reverse read, like that. So you get to see the reads. Can you see some regions that are very minimally covered only in this particular process? For example, you see here that green line right out here. That is, how many times has it been covered? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Less than 15 times. So you know that. So what do you call that? Once it is covered only 15 times, what is the terminology that is used there? Depth is 15x. Perfect. Now you can see out here, for example. So this is one region where if actually I can scroll down also, but this is an image, so I cannot. But you can see this is having a very high depth, right? So this this graph that you can see, what is that showing? That is showing the depth. When you look at the height of it, that is showing the depth of sequencing. When you go horizontally, obviously that is also showing you the coverage, right? So you know that some of the regions are not covered at all. What else can you see here? You know, whenever you see such a color coding, so by default it is gray. Whenever you see such a color out here, so that color denotes the presence of a variant out here, So you can see there are two colors out here. And what else do you see here? It is equal, right? Nearly equal. So one color is forming half of the, half of the reads are having one color. The other half of the reads are having a different color altogether. So if you just look at it, this particular region, so there, you know, the depth is 127. So that region has been interrogated 127 times. Of that 127 times, 65 of them showed A and 62 of them showed G. What do you mean by this? This is most likely a heterozygous variant which is present at that particular location. And you can also see what is the location, that is chromosome 4105. This is a genomic loci. So this information you can get by visualizing your BAM files in the genomics viewer. Obviously, I mean, you can imagine it's not always possible for us to visualize each and every variant for this. We can also get the same information in a tabular format also at some later stages of your analysis. But you can see out here. What else can you see out here? Can you see here in this one, there is a clear-cut fall in one particular region in the genome. So, you know, everywhere you are getting good number of reads, but only for this particular base out here, the number of reads really dropped. Why did it drop? When you're comparing with the reference sequence, you know, you can always look at the reference sequence here at the very bottom. When you're comparing it to the reference sequence, you see that there are actually 105 reads out here. All the 105 are Gs. But apart from that, there is deletion in almost 93 reads also out here. There is no particular, I mean, there is no nucleotide for 93 reads in that particular position. What do you mean by that? It is again a heterozygous deletion of a particular base. So most likely, originally it should have been a G, and but in half of the reads, that G is deleted. So, you know, if this is in the coding region, what will it lead to? It will lead to a frame shift, right? Because you're having one G that is getting deleted, it will lead to a frame shift deletion. Depending on the position of in the protein, etc., it might be deleterious. It can affect the protein function in at least half of these conditions. So that is something. Yes. Yeah. Yeah, I have a picture here, I think. Somewhere here. Yeah, right this. Yeah, right. So like this. No. So basically what happens now, your aligner will still align it across that particular region. Okay. And aligner will tell you that, okay, I tried my best to align it, but there is a gap. That is how it works. Yeah, Alena will do that. So Alena will not go for exact 100% matching, but it will try to do as much. So you can you can increase the sensitivity and also like how much matching you need, how much percentage of matching you need, and all those things can be there. So basically, yeah. So this is how you visualize a BAM file. Now, my only problem out here. So why do I need these numbers, right? Because these numbers are important. How many reads I get when it comes to each basis, especially when there is a variant. Because here I can see that 51 and 49, which means that it is a germline variant which is coming from which is actually equally equally present in both the reads. So that number, that percentage is also important, because if that percentage is slightly off, for example, if I'm getting 80 and 20, one base is 80, the other one is 20, then I'll be in a fuss, like whether it is germline, whether it is somatic. Most likely I will be going more towards somatic in that situation like that. So these numbers are important, which means that, when you're actually doing your sequencing, you should be aligning unique reads that are coming from your sequencer. All the reads should be unique. When I say unique, what do you mean by that? So if you see all these reads out here, each of them, they have a different starting point, they have a different end point, right? So this read has a particular starting point and end point. I don't see any other read out here, at least in this picture frame, which has the same starting point and the same end point, right? So if I see this gap, it's all, there is no other read that is starting at that exact point. Maybe you can say this one is starting at that exact point, but then it is not ending at the same point. It's ending somewhere out here. Are you getting what I'm trying to say? So each of these reads are unique when it comes to their starting point and ending point. So that is something. Yeah, you get a higher depth, not only higher depth. Imagine if you have a variant, this proportions will alter, right? Now you cannot tell that your variant is present 50% times compared to the other one. So that numbers will get distorted. So it will actually give a false sense of depth, correct? And it will also lead to errors during interpretation. So that is why we have to remove PCR duplicates. Okay, we have to remove the PCR duplicates. So how do we do that? That is with the help of a tool which is known as, again, there are n number of tools for this particular process. This step is known as marking the duplicate. So rather, we don't remove the duplicates, we mark it as a duplicate and keep, we keep a flag. Okay, this sequence, it is a duplicate sequence which is coming from a particular, it is a PCR duplicate which has come. So this process is called marking the duplicates, and later when you're analyzing it, you can ignore all those reads that have been marked as a duplicate. So this process is known as marking the duplicates, and we use a tool which is known as Picard, this one. So that is used for marking the duplicate. Okay, that is with the help of Picard. So that is basically how we do the marking of the duplicates. The next thing that you are going to do after marking the duplicates, now let me just quickly revise what exactly we did till now. So we aligned it to the reference genome, we sorted it, converted it into a BAM file, and we marked the duplicates. After we are done with all these steps, we can do a QC, another round of quality control, to know how good your alignment and mapping was. How good your alignment and mapping was. The tool that we generally use for this particular process is known as, which we use commonly, is called QualiMap. QualiMap. I'll show you the results of a QualiMap output. So let's say, is it here? So I can input multiple samples or a single sample out here. So here I did, I think, some 25 different samples altogether. Doesn't matter. So yeah. Can you see here? So this is one. I'll show you. Let me open one more file simultaneously for you. Right here. Okay, two different files. Okay, this is one file. What we generally look into here, you get two important information right out here. One is this thing, mean sample mapping quality. This is also something like your Phred score. It's a negative logarithm, and which means that what is the probability of your map reads having an error. So again, when you say 56, it is approaching something like 60. 60 means 1 in 10 raised to minus 6, 1 in 1 million errors. So that's the probability of a wrong alignment. Yeah, generally we get it because with Illumina, if you start with good quality data, because you're actually sort of removing a lot of your ambiguous reads and everything, filtering out previously itself, right? So you start with good quality scores and everything, so you get good values like this. The other thing that you are seeing, so this is an exam file, okay? So that is why we have this. Yeah, right. No. I mean, we have removed some bad reads, that's it, and adapters. No, I mean, the process is similar. Pipeline is similar. Rather, for most of it, the pipeline is similar. Only when you look at the final data, I mean, it will be different. There are a few steps where it slightly diverges, but mostly it is similar. Yeah, all right. So can you see, actually, I told you a lot of people get confused between coverage and depth. So here in Collimap also, the people who designed it, they also got confused between coverage and depth. Actually, this is depth, okay? What is a mean sample depth? It means that, the depth is 150 times on an average. So remember what is the target for an exam? It is 100x. So here it is way more than that. So it's almost 150x data that we have, okay? WGS is 30. I'll show you the WGS data also. So this one. So you can see the sample coverage is 29.9. The whole genome sequencing. There also the mapping quality is good. See the difference in the GC content. Okay, the human genome has 40% GC, but because when it comes to exomes, you are doing a selection. The GC content is closer to 50, at least for this exome, because we are picking up only the exonic regions, right? So that's why you see a difference here. GC content is close to 50. So this is what you see here. Also, you can have a look at, if I scroll down, you see, for example, something like this. So what is the coverage across your reference sequence? So you can see throughout most of your reference sequence, the coverage is relatively good. No. I mean, it's actually a arbitrary thing. So you see, I mean, it tells you something like position in the references. It's like an artificially generated map. So this axis you have like, I mean, each of them is a sample, okay? So each of this color is a sample. I have 25 samples. That way it's looking very crowded. But if I show you somewhere here I have only 2 samples. So you can see here. Yeah, I mean, so this could be multiple regions across the genome. One problem is obviously your centromeres and telomeres will not be covered. One problem. Repetitive regions, you will see a drop in the coverage. And remember, we also use those alternative contigs, right? So you had your main chromosomes and then a lot of alternative versions of your chromosomes. When you saw the yeah, so those will all appear as blank in that particular process. But we look at it, and especially rather than more important than graphs, many times we have a look at these numbers because it has happened to us once we have aligned it. I mean at least once we got for a few files a very low alignment quality, especially for a whole exome. Which means that when they did this process, probably they messed up somewhere. The capture was probably not great, or probably the purification was not great when they did that. So once it went into sequencer, I mean, we didn't get the expected regions as an output. I mean, so there also remember your FASTQ and all those things will look perfectly fine, but until unless you align and see what exactly